So then CRISPR-Cas-based gene editing that you have all heard about, it's very popular. So with this technology, actually the gene defect, the mutation which is there in your gene, it will be corrected right there itself. In your every cell, this technology is designed in a way that in every cell, it can correct that defect. But it has not yet reached a clinical stage. It is still in testing and preclinical stage. The advantage is that it can correct the mutation and restore the expression of full-length dystrophin. And it will also be a one-time treatment. So once you take it, then all the mutations hopefully will be corrected, and that you'll have a lot of dystrophin made in a normal situation in each cell. A disadvantage is that when you are editing the gene, that means you are there was a mutation and you are correcting it. So you want only the mutation which was in dystrophin to get corrected. But this machinery CRISPR-Cas, sometimes unintentionally, it can go to other targets, other genes also, where it is not supposed to touch. And that can have off-target effect, they say. That means instead of dystrophin, it is tackling dystrophin. At the same time, it is going to some other gene also. And that can have disastrous effects. So this is a drawback of this technology, which they are trying to improve it. And the cost for this also is going to be high. Then instead of DNA, we can also use messenger RNA for therapy. And messenger RNA is the one that we need to read into the protein, the translation step. So in messenger RNA therapy, we put a copy of the messenger RNA. Difference between DNA and messenger RNA is that DNA is a stable molecule, whereas messenger RNA is unstable. It is broken down in the cell after some time. So the advantage is that since messenger RNA is not stable, it gets broken down. So it is transient, and that helps to control the dosing. So as I said that in the gene therapy, one dose only is given, so it has to be correct. If it is too high, it can be toxic. Too low, it won't have effect. In messenger RNA, that is not a problem because you have to anyway give it repeatedly because it gets degraded. Production is also simpler than DNA, so it could be that it could be less expensive, but we have to see. It is safer because you are not touching the DNA. So whenever we do anything at the DNA level, there's a risk of genotoxicity. That means it can lead to cancer. So this risk is not there with messenger RNA. Disadvantage, it is unstable and it breaks down. So that means it's not one-time treatment. We have to keep on going again and again for treatment. Then the technology has still to be improved so that we have sufficient amount of mRNA should reach the muscle. So you have all heard about the COVID mRNA vaccine. So actually it is the same technology. In mRNA vaccine also we are giving the mRNA for COVID, and then it is making the COVID protein, which is helping, which is like the vaccine. But the difference is that when we want to use the same technology for DMD, we have to make a large amount of protein. For vaccine, we need very small amount of protein. So that's where the technology has to be improved. We have to make sufficient amount of protein so that it will actually have an effect. If the protein is very small in amount, then it will not have much effect on the muscle function. So these are the some of the technology, some of the steps in this which are being optimized. The other is the anti-inflammatory drugs, which are now the standard of care, and most of the patients are taking anti-inflammatory drugs. So they have been of help to patients, but they also have side effects. So people are trying to come up with new anti-inflammatory, which are not which are synthetic steroids, and they have less of side effects. So this vamorolone is one such example, where a clinical trial has been done with 46 boys, and they found that it is as effective as glucocorticoid and it has less side effect. So in glucocorticoid treatment, the growth of the child slows down. But here with vamorolone, the growth was not impaired. So it's possible that in future we will have better and better non-steroidal or synthetic steroidal molecules, which will have less of side effects. Then there are molecules like myostatin. Muscle tissue, when we are growing or when the muscle is damaged and new muscle is made, so it has to be regulated very properly. You don't want to have too much of muscle, and of course you don't want too less. So myostatin is a molecule which regulates how much muscle has to grow in the body. So myostatin stops the muscle from growing if too much has already grown. Now what they're doing is that if I inhibit myostatin, then muscle growth can be promoted because it has a negative effect on muscle growth. And there are drugs which have been tried. Now what they found is that with these drugs developed by Pfizer and Hoffmann La Roche, was gain in muscle mass, but the strength did not go. So even though muscle growth was there, but it was not, didn't have strength because dystrophin is not there. So therefore, it is thought now that myostatin could be used in combination with other therapies where dystrophin level can be increased, so that then myostatin can have better effect. Another molecule is utrophin, which is similar to dystrophin. But the thing is that utrophin is not made in all the muscle cells. It is made in the neuromuscular junction. So they have, by research, they have found out that there is a protein called GAL-GT2, whatever, don't bother about the name. This protein, it helps the utrophin to express in the myofiber itself. So if this protein is there, then utrophin can be expressed in the muscle cell. So now it is being tried that through AAV vector, this GAL-GT gene is being delivered, which will be expressed in the DMD boys, and then it might promote utrophin expression, and that could overcome the defect of DMD. This is being tried. Okay. Now, this is as I told you, that a lot of research is going on in many avenues. So I have just picked up little examples of various types to give you a flavor of so many things are happening. This is an example of research which is very encouraging and very good. So this is so yeah, the other thing is that, we have scientists who are working very closely with the pharma companies. This is of course all happening abroad, not in India. Now, what is happening is that a lot of the scientists themselves are opening the company. Okay. So like the SATELLOS Bioscience is a company in Canada. It is opened by this scientist, Dr. Rudnicki, who has done this work also. So this is a very good thing that, the scientists first, after many years of research, they're coming up with something, and then they're opening a pharma company to develop the drug also. So various such examples I will tell you. So SATELLOS, what have they done? So I have to explain a little bit. Now what happens that, muscle, whenever there's muscle damage, let's say due to exercise or any other reason, so then muscle has its own stem cell, which is called the satellite cell. The satellite cell, so this is the satellite cell. Satellite cell will divide, and then when in the normal situation it divides in a way that is called asymmetric. Means the two cells which come out are not the same. One of these cells will again become a satellite cell, and the other cell, this one in green, this is the one which will become muscle. This will get converted to new muscle. Okay. So this is a very important point which Rudnicki's group, they have shown that this asymmetric division, which is very important for muscle regeneration, this is not happening in DMD. Now in DMD, where dystrophin is not there, the satellite cell actually division is symmetric. Both the cells are like more satellite cells. So when satellite cell divides, it gives rise to more satellite cell, and this green one, which will get converted to muscle, this is not formed. So that's the reason that muscle is not forming in DMD. So this observation Dr. Rutiki's lab made, and then they said that suppose we can induce, in this DMD situation, we can induce this asymmetric division, then what happens? So they have tried out various drugs, and they actually came up with some small molecule drug which can induce this asymmetric division in the dystrophin in DMD. And then they have used it in MDX mice, and it's a 4-week drug treatment they found with this drug. It has really improved the running speed of the mice and also formation of functional muscle fibers. So their initial results are very encouraging, and they've also tested in dog model. But to reach a stage where they can do clinical trial, they probably need another year to do more toxicity studies, etc. But this approach is very interesting, and they're coming up with molecules which can possibly help to generate new muscle right in the DMD situation. Then another one is this Edgewise, another company, Edgewise Therapeutics, where so here the technology which they are developing is based on this thing that, I told you that the dystrophin protein, it connects the inside of the cell with the membrane. So it gives a protection. So whenever there's a contraction of the muscle cell, the membrane remains intact. But in DMD, when there's a contraction, the membrane ruptures. And that leads to all kinds of things like calcium influx, and ultimately the cell dies. So their logic is that suppose we can reduce the contraction. So this is damage is happening because of contraction. But contraction needed for muscle to function. So if you reduce contraction, how the muscle will function? So what they're saying is that let us have a contraction just enough for muscle function, not extra. So reduce it to the point where muscle can function, but this damage not happen. So they have come up with an inhibitor of contraction, and they are trying this inhibitor, this molecule ETG5506. They have tested it in MDX mice and in dog model, and they are finding that it protects the muscle from membrane disruption, and it reduces the diaphragm fibrosis. And in dog model, it increased the daily activity of the dog. So they are getting encouraging result. And they have also done safety study with healthy volunteers, and it is not toxic. So now phase 1 and 2 studies are being planned for both BMD and DMD. Then another therapy which is being developed is based on cardiosphere-derived cells. This is by this company called Capricorn Therapeutics. So cardiosphere-derived cells are cells which are from the human heart muscle. And these cells have their ability to secrete molecules which can promote regeneration of tissues and reduce the inflammation. So this is they have got these cardiosphere cells from human heart muscle and then infused intravenous every 3 months for 1 year. 4 injections they have given in a phase 2 clinical trial with 20 DMD boys, and the results have been encouraging. So this technology is also being tried out with larger cohort of patients. So now about some more cell-based therapies. So we keep hearing about stem cells, and stem cells are really promising. But we have to be careful that just because we have a stem cell and it can get converted to muscle or any tissue, it doesn't mean that it will work, because the efficiency it may get converted to muscle, but let's say that the efficiency is very, very small. The percentage of cells which are getting converted is very small. It's not going to have any effect, positive effect for the patient. So therefore, with cell-based therapies, we have to understand that, what are the limitations and how to overcome. So you have heard about mesenchymal stem cells and induced pluripotent stem cells, embryonic stem cells. All these cells are actually possible for us to use to get converted to muscle. But the efficiency so far has been found to be poor for embryonic stem cells. Also, there's a hazard that they could become cancerous because they are highly immortalized cells. Now, the cells can be obtained from the patient themselves, then it is autologous. And if they're obtained from the patient themselves, then they will have the DMD mutation, the dystrophin. So it has to be corrected. So the advantage of autologous is that you won't have immune rejection because the cell is from the patient themselves. But the disadvantage is that you have to genetically manipulate the cell, remove the mutation, and that is not very easy. Because manipulation in the lab, you have to make sure that you have not introduced any other problem because of the manipulation, and because it is done for each patient separately, so it will be expensive. So on the other hand, if you take cells from healthy donor, that is allogenic, then the problem, the plus point is that one healthy donor can give cell to a large number of patients. So it is like off the shelf. You can order for it and you can get it, and cost will be lower. But problem is that immune rejection would be, so you have to do immune suppression. So what are the cell-based therapies which are promising right now? So one company which is there is Beta Therapeutics, which is really doing very lot of work on cell-based therapies. And again, it is done by a scientist who have now, of course, Beta Therapeutics has taken over their technology, but it was initially developed and being developed by these scientists. And as I told you that the challenge with the stem cell therapy is that the efficiency of differentiating into muscle is low. So Vita is really trying that to get enough cells so that they can differentiate into the muscle cell. And there are various products like VTA110 is an allogenic therapy for iPSC based. Then VTA100 is an autologous treatment for LGMD. And VTA200 is a universal stem cell. That means here the cell has been modified so that it will not be immunogenic. So they are coming up with various kinds of these cells, and we have to wait to see that what kind of therapies will be best for us. This is in Poland, again by a scientist couple who have shown that they are using chimeric cells. So this is a technology which they have developed for muscle. What they're doing is that they're taking myoblast, that is a cell which will get converted to muscle from a normal human and also from a DMD patient. So two cells, one is allogenic from the normal human, one myoblast, the other is autologous from the DMD patient. So these two cells here, they are fusing them. So it is getting fused into one. Now this cell is one that it will make the normal dystrophin from the normal myoblast, and it will also have the signature of cell from the patient, because now this cell will appear to your body as your own cell, because the patient marker is also there on the cell. So with this therapy, the advantage is that you can make dystrophin at a normal level. At the same time, immune rejection will not be there. So phase 1 clinical trial has been done with 3 boys to check the safety of this, and so it is going to be tested in more boys, but it looks like quite promising that, here you're overcoming immune rejection by making a fused fusion cell, a chimeric cell. So these are some of the things that I wanted to tell you, the new kind of approaches which are happening. All of them are quite encouraging. So just to summarize, therapies for DMD are being developed at all levels. DNA level, we have micro and mini dystrophin trials are going on. Then RNA level also is being worked on. Then we have exon skipping and nonsense suppression so that the incomplete protein or the frame shift problem is solved and we can make the complete dystrophin protein. Then muscle growth, if we can induce by myostatin inhibition or utrophin overexpression, that is being tried. New anti-inflammatory drugs with less side effects. Then satellite cells, asymmetric division, how to induce, that is being tried. Membrane damage, how to reduce, that is being tried. Cardiosphere derived cells is another method to induce regeneration of muscle. And stem cells to improve the ability of stem cells to differentiate into muscle. And finally, the chimeric cells, which can overcome the problem of immune rejection. So all the therapies that I've told you, none of them is actually perfect today. They all have plus and minus points. So, but a lot of work is going on to improve them. And maybe we have to think also in terms of combination therapies. Just one therapy alone may be less effective, but if we can combine, say, two therapies, then it could be more effective. The problem is also that even when the therapies come, they are very expensive and the cost is so high that we will not really be able to afford them. So therefore, it is important that we, like as patient groups, we have to tell the government that, we need to make and test drugs in India. That's the only way that we can have affordable therapies for our patients in India. And thank you very much. So I'll be happy to answer any questions.
Yeah, ma'am, one question came out is like, how is the age affect this, how is correlate with the new medicine? So for many cases, so we have the few of the DMD warriors, so with more than 20 years age and all. For them, what will be the hope for the new medicines?
For the older patients?
Yeah, for all the older persons.
Yeah. So that is that's where I think the cell-based therapies could be more better for the older patients, because once a muscle is already damaged, then even gene therapy will not be that effective because it will not improve the damaged muscle. But if you can introduce, say, cell-based therapy, then it is like a organ transplant, So you can give, new muscle, and if it works out, then that could be quite effective for older patients.
Okay, thank you, ma'am. Another one question is like, for example, one patient tried with a gene therapy or exon skipping, and later after a few years, so maybe another better gene therapy came. So he can utilize that new therapy, or once we applied that exon skipping, they could not go for the other gene therapy or something, if there is any restriction.
Exon skipping and gene therapy should not have any danger, because for exon skipping, you're taking an antisense oligo. That's the most common, isn't it? For exon skipping, you are taking a molecule from outside, antisense oligo, and you're taking repeatedly. It's not one time, you're continuously taking it. So along with that, you can take gene therapy. But the thing is that if antisense oligo is working for you, suppose it is working, then you don't need gene therapy, because that is already effective. But if it is not effective, then definitely you can go for gene therapy.
Okay, so there is no restriction in choosing the newer therapy.
Yeah, the restriction is only that if you have to repeat gene therapy itself,
Okay.
like, or anything to do with AAV vector. So if, either it is CRISPR-Cas editing or it is gene therapy where we need the AAV vector system. There you have to be careful because AAV antibody we already have. Now, if you have taken AAV for one therapy and you have gone through immunosuppression, then for one more of the same could be dangerous.
Okay. From the audience, so any other questions? You can ask the questions in Malayalam also. എന്തെങ്കിലും ക്വസ്റ്റ്യൻ ഉണ്ടെങ്കിൽ മലയാളത്തിൽ ചോദിച്ചാലും മതി. Let me check any other questions in chat.
Sunil has something.
സുനിൽ സാറിനെ ഒന്ന് അൺമ്യൂട്ട് ചെയ്യോ?
Hello. Hello, madam. Can you?
Yes, Sunileta, proceed ചെയ്യോ?
The Krishna Maharajist. Madam, thank you for your great speech. It is very informative. And madam, my question is: after hearing, I think that the stem cell therapy is the most promising and cost base. It is affordable. I don't know whether I am correct or not, but it is just my thoughts. I think if we mix with the stem cell therapy with the other therapies, can it produce the great result? Can it cure the DMD completely?
I'm sorry, actually your voice is very faint. I wasn't able to hear. Can you tell me, Krishna, what he is asking? I can't hear Sunil's voice. Oh, I can't hear.
I will repeat once again. Okay. Okay. Now, can you hear now?
Can you speak very loudly? Yeah, now I can hear. Yes.
Okay, madam. First, thanks for your great speech. It is very good and very informative. Then my question is, after hearing your speech, I think the stem cell, if the stem cell therapy is the most promising. I know it is a very premature stage, but after hearing, I think that stem cell therapy is the most promising. My question is, if the stem cell therapy is combined with the other therapies, is it create a magic result? Is it cure the DMD completely? That my question is, combinations with the other therapy, like the gene therapy or UTROPHIN upgradation plus stem cell therapy will make the non-ambulatory to ambulatory.
I mean, the term you're using, magic, is very dangerous because there's really no magic in this. So as I told you that the company Vita Therapeutics is working quite hard to improve it. Okay, so the technology is there, and when in a scientific paper you read it, you feel, oh, problem is solved. It's so good. But actually, when we apply it to the patient, is going to be very minimal. That's the problem. If the efficiency is minimal, then all of these therapies have some issues also. Okay, because if you transfer cells into a person, is not such a harmless thing, and it can become cancer also. So the efficiency of converting to muscle should be high. Otherwise, why take so much of risk? So these are, I agree with you that it is very promising, no doubt about it, very promising. But it's not that it is already there. Okay, it is being optimized. And I have not talked about other papers. There are some other groups also doing where they are looking, they are come up with some molecules. Those.
Yeah, one question on India perspective. So, what's our hope? So, in which area so we can get some medicine, especially considering that the kind of research happening in India?
Yeah, so that's very important, that what are we able to do in India? So we are doing very less actually compared with what we should do. But the thing is that at least some beginning looks like is being made. So this COVID, during COVID, the mRNA vaccine has been now designed by Genova Pharmaceuticals in India, and as I said, that to make the vaccine, you don't need too much of a protein made from each messenger RNA. But if we have to use it for genetic disorder, then we need to make much more protein. But it is a possibility. And Genova, if they are also attention to genetic disease possibility, because they already have the infrastructure. They know how to make messenger RNA, how to make it into protein and so on. So that possibility is there. Then also there are some companies coming up where they are doing AV vector protein. Thing is that to use as gene therapy. So that is also now happening in India slowly. And cell-based therapies are also, initiating. But I don't see any big company in India coming up with it. It's not. They are all small ones. But maybe we as patient groups, we have to talk to them and to encourage them and show them that they have a big market. All patients are here. So if we can make a sort of consortium where patients along with the doctors can then talk to pharma companies, even if it's a small company, but if they are willing to dedicate themselves, then we have to make a beginning that way. It hasn't yet happened in India, but I think like your mind and your
Correct, correct.
organization very
Actually,
encouraging that.
we are trying our best. So we are connecting with other patients' organization as well as the parents' organization, and collectively trying to maybe negotiating with the pharmaceutical companies and all. So definitely we may require so ma'am support as well. So maybe that in the later point of time we may require your support as well. So we are expecting all the support from you as well.
Yeah, sure. We need the same thing. We are all
Yeah.
in the same boat. We all need, if some company can come up to do either any of these therapies, if we need a company to dedicate itself, as we are finding in US and in other countries, that the company along with scientists is fully dedicated to a technology. We need that to happen in India. Then, along with patients, we are working closely, then things can work out.
Yeah, thank you. I know that this week you had some health issues, and but still you are able to make this session wonderful session and able to cover that what is currently happening in the DMD area and what is expected things to be happen. So thanks for providing a wonderful session. Thank you very much, ma'am. And I'm personally, personally as well as that the mind's point of view, I am giving our sincere thanks to ma'am for this wonderful session.
Thank you.
No, not an issue because we know that we all are connecting remotely, that technological issue. So that's okay.
Yeah.
Thank you very much.
Hopefully we meet in person next time.
Yeah, definitely we'll visit. Yeah.